The treatment, made out of nanoparticles molded from the cow pea mosaic infection, an infection that taints dark peered toward pea plants, showed amazing progress in further developing endurance rates. Also, smothering the development of metastatic growths across different disease models, including colon, ovarian, melanoma, and bosom disease. Comparative results were likewise seen when the treatment was directed to mice whose cancers were precisely eliminated. The discoveries were distributed as of late in cutting-edge science. The new review expands upon past examination by the lab of Nicole Steinmetz, a teacher of nanoengineering, overseer of the Middle for Nanoimmunoengineering, and co-head of the Community for Designing and Malignant Growth, all at UC San Diego. Steinmetz and partners have been utilizing cow pea mosaic infection nanoparticles to set off the resistant framework to battle disease and keep it from spreading and repeating. In early examinations, the methodology included infusing the plant infection nanoparticles straightforwardly into cancers to invigorate an insusceptible reaction. Despite the fact that the infection is non-irresistible in well-evolved creatures, the body's safe cells actually remember it as unfamiliar, setting off a powerful safe response against the current cancer, as well as any future growths. Presently, Steinmetz and her group show that the plant infection nanoparticles needn't bother with to be infused straightforwardly into cancers to be successful. Regulating the nanoparticles foundationally moved along. Endurance rates and restrained metastasis across different malignant growth types. Here, we don't treat laid-out cancers or metastatic illness. We keep them from framing. We are giving a fundamental treatment to awaken the body's insusceptible framework to dispense with the sickness before metastases even structure and settle, said Steinmetz. To make the nanoparticles, the analysts developed looked at pea plants in the lab and contaminated them with cow pea mosaic infection. A huge number of duplicates of the infection were developed and reaped in the type of ball-formed nanoparticles, which required no further change before use in tests. Nature's strong nanoparticles, as delivered in dark, peered toward pea plants, said Steinmetz. The specialists tried the viability of the treatment in mouse models of colon, ovarian, melanoma, and bosom malignant growths. Mice infused with cow pea mosaic infection nanoparticles, and then at that point tested with metastatic growths 7 days after the fact, displayed better endurance rates and decreased cancer development contrasted with untreated mice. In any event, when tested with new growths a month after the fact, treated mice showed comparable results. The analysts are especially amped up for the treatment's viability post-medical procedure. In one more arrangement of tests, managing the nanoparticles after careful evacuation of cancers brought about better endurance rates and diminished cancer regrowth in mice. Regardless of whether you carry out procedure to eliminate the growths, no medical procedure is awesome and there is outgrowth of metastasis assuming no extra treatment is given, said Steinmetz. Here we utilize our plant infection nanoparticles after medical procedure to help the safe framework to dismiss any lingering illness and keep flowing growth cells from metastatic cultivating. We found that it works extremely well. The objective is to prepare for clinical preliminaries. As the exploration advances, the group will lead well-being studies and investigating the therapy's viability in pet creatures with malignant growth. Future investigations will likewise zero in on understanding the components fundamental the resistant helping properties of cow pea mosaic infection nanoparticles.